Right, here we are at our tour destination, Fordham Hall. This is one of the older buildings of the medical school, and it's named after a former chancellor. And yeah, he was a scientist as well. And you know, he gave a lot of money and support to the university, of course. So that's how you get your name on a building. Let's see, this building is home to several labs in biology, genetics, those sorts of sciences. And so the lab that we're going to see today is the home of Dr. Kerry Bloom, who's the chair of the biology department. He also works on genetics research that you'll learn about later. Let's go inside. Dr. Bloom, how are you?
Good, how are you, Thomas?
Good.
Good.
So, what are we going to see today?
My lab.
All right, let's go take a look.
Okay.
So, Dr. Bloom, thank you for having us today. One thing I wanted to ask you to do is, introduce yourself, tell us how you got to UNC, and tell us something that you really love about UNC.
Okay. Wow. So I'm possibly one of the people that have been at UNC the longest that you're talking to. I came here in '82. I know, it seems crazy. I just basically knew nothing about science and knew nothing about being a scientist or anything when I applied. I was pre-med all the way as an undergraduate, for those undergrad pre-meds out there. And I sort of started figuring out I liked science when I was in college doing experiments in a lab because I wanted to get a letter of recommendation. And to make a long story short, I fell in love with science and I got to UNC because it was where I had a job. And I've been here ever since and I love it.
Cool. Where'd you go to grad school?
I went to grad school at Purdue.
Okay.
And I did a postdoc out in Santa Barbara, California.
Oh, awesome.
I really, really love California right now. I am so happy we're in Chapel Hill and not California.
Indeed, indeed.
For many reasons.
Yes.
Yeah.
Yeah. And so what's something that you really love about UNC?
I would say the one thing, if I put my finger on, that has kept me here over all the years is collegiality. We often ask faculty, when we're recruiting faculty, or even grad students for that matter, we ask them, where do you see yourself in 10 years? And the answer is, of course, you don't know. Mm-hmm. But what you can answer is that not what I'm doing now, right? Because if you're doing what you're doing now in 10 years, you're not at the forefront of whatever you're supposed to be doing. Mm-hmm. And being able to sort of switch fields around and explore new areas of research essentially means you need to have people to talk to mm-hmm. and to help you learn. You talk about lifelong learners. I know it sounds so cliche, but I really feel like I'm a lifelong learner, and it's because of faculty in physics, in math, in computer science, in chemistry that have really enriched my time here, which is easily the reason I'm here. People are very open about sharing resources and ideas.
Yeah, so it sounds like you're saying that just because you love one particular discipline, or that's where you start out, that doesn't mean that that's the only place or the only discipline that you should be thinking about.
Exactly.
Yeah.
Yep.
Awesome. So tell us a little bit about your research and why it's important.
Okay, so the simplest way to describe it is that my research is on how cells manage to divide their DNA equally to mother and daughter cells at every cell division. We are a tribute to the fidelity of that process. Depending on how you count cells, we're a couple of trillion cells, and the error rate for our chromosomes being distributed accurately is insanely low. We are a tribute to how accurate that mechanism is. Nothing we make, by the way, no matter what computer program, algorithm you make, has the fidelity that we have somehow managed to achieve in cell division. So I'm interested in how that works, mm-hmm. Which means I'm interested in how the DNA actually behaves in the cell And so many of you are probably familiar with DNA from the perspective of the code. The code obviously is the sort of the new kid on the block, so to speak. Everyone knows about the code, everyone knows about gene testing and da-da-da. There's another way to look at DNA, and that is from the perspective of a long polymer chain. So I'm interested in how that, because the other way to look at DNA is, you may have heard this little thing, but if you take all the DNA and all the cells of your body, how long would it stretch? And basically it's to the sun and back. It's some unfathomable number. And so how do we package that DNA in a way that makes it work, basically? And so I'm interested in the packaging side of it and the separation side during cell division. And so I'm interested in it from the polymer characteristics in terms of how that. So I do have a little demo that I'll show you in a few minutes, how we think about it. But yeah, so I've sort of gotten into polymer physics. You'll see we do a lot of microscopy because I want to watch the DNA move around, and understanding how it moves around is obviously a key part of how it segregates.
Right, and that's really interesting because, you know, I'm a biochemist and I think about proteins, and for those, you know, sequence is really, really important for structure. But for DNA, you know, the sequence doesn't matter that much for structure.
Exactly. Yep.
And so you have to think about slightly different things to think about the physics of how that works. So that's really fascinating.
Yep.
All right, so why don't we go take a look and see what you're working on?
Okay. One thing we do is we build microscopes.
Oh, you build them?
We build them.
Okay.
We build them. If you take us, if you zoom in here, you basically see an objective, and you see a bunch of equipment essentially that's all wired in with different Allen bolts and things, and if I take this top off, you see we have all of our lasers shooting into mirrors. Mirrors are directed into an optical fiber that comes into the microscope. So these are handmade microscopes to give us certain features that we require.
I see. So once you all build these are pretty much one-of-a-kind instruments that you couldn't possibly find anywhere else.
That is correct. Or if you found them someplace else, they would probably cost $5 to $6 million.
Ah, okay.
Yeah. So we save a ton of money. Literally by building our own. So we'll walk around this way.
Okay.
And I'll try to show you one where we actually have some cells on a microscope. Thomas is already here doing it. Awesome. This is what I was going to show you. So these are our yeast cells. Can you even see that? And those little green spots are spots of DNA. Yes. Oh, awesome. So we watch those spots move around in real time.
Oh, nice.
What you can see without seeing too much is that everything is computer powered, driven, and analyzed images off of the computer.
I see.
Can you? Yeah.
I got it.
You got
Awesome.
it? Hi.
Yes. Hi.
So the nice thing is it allows us to look at different proteins live. So we have the microtubule organizing centers labeled with our spindle body protein in red, RFP. Of course, everything is just black and white, and it's pseudo-colored. And then we're interested in the nucleolus and how that's organized. And so we have a protein CDC42 labeled with GFP. And then you can overlay it, and you can do live cell timing or just take individual population pictures.
Very nice.
But that's what we do in here.
So what is this?
So this is actually a 3D printout of what we call the centromere region of the chromosome. So we were talking about chromosome segregation. These are the microtubules, these guys, these long rods. And when the cell divides, when the cell decides to separate the DNA, the microtubules separate apart. They pull the DNA with it. This will go to one cell, and this will go to the other cell.
I see.
And this is part of the structure we think that's important in how that whole
I see.
thing works.
And then the rest of the chromosome, would that extend up this
exactly
way?
the rest of the
Okay.
chromosome would extend up and
Okay.
down.
Very cool.
This is my this is my representation for what DNA looks like in most of your cells, which is how does this thing work? And this is the demo I was going to show you. So do you see? So basically, this is a chain of paper clips. And it turns out this is a really good model for DNA. And you see this little piece of green tape? That indicates how we label the DNA. So we literally have a strand of the DNA labeled amidst the whole long chromosome. And you can watch this thing wiggle around, and we are interested in that wiggling as to how the chromosome works. So this is a movie of showing you that green spot on this paperclip. And now what you can see is that, yeah, you just saw it. If you catch it, I'll run it again.
Yeah, run it again.
DNA is stretched out between the two daughter cells. In this case, they have a mutation, and so the DNA collapses, and then you see the two green blobs into the two daughter cells. So we basically try to analyze thousands of these movies and glean some information about how the chromosomes are actually working.
And so talk a little bit about time scale. How long is this happening?
This whole thing is probably happening about 8 minutes, 8 to 10 minutes. So in terms of like, if you're thinking like physics times, this is actually very slow. So things it's like swimming in tar inside the cell. So it's, the atoms are moving fast, but the molecules are moving really slow because it's a really, really thick environment that they're trying to move in.
Now, earlier you mentioned that you work with Saccharomyces cerevisiae.
Yeah, so that's what this is Saccharomyces cerevisiae, these large budded cells.
So why is that your model of choice?
That's our model of choice because in spite of all the brouhaha about CRISPR and gene editing and all of this cool stuff we can do, there are no ethical issues in working with cerevisiae, and everything is really fast. So we do gene editing in like 2 days. It's just really fast. We can manipulate chromosomes, build chromosomes, put in all kinds of different mutants. And so it's just a very facile genetic organism. I'm going to sort of give you a snapshot as to how we came about to build this three-dimensional model of the chromosome. And so that's shown here. And what you're going to see is 2 strands of DNA. Can you see that? Yeah, 2 strands of DNA. This probably takes, I'll cut a sound that my undergraduate put in. I'll speed it up a little bit.
So did an undergrad help put this video together?
Absolutely, an undergrad. So my undergrads do a lot of the computational analysis, it turns out. Yeah, you got a good shot of it. So now you see the loop growing, and you see it making multiple loops. And now you see we're doing some DNA origami here. So somehow the cell does DNA origami, and it brings in a number of different chromosomes.
Whoa.
Yeah, and so we think this is what's happening inside the cell. If I now take this and show you what happens from the physics side, that's the 3D printout. That's the mathematical model. And now what you can see is the strands are wiggling and wiggling like crazy. This gets back to the wiggling of these DNA chains. And it's from thinking about how these chains wiggle around and the structure they form that helps us understand chromosome segregation.
Very cool. And so thinking about, looking at chromosome segregation, what types of issues do people have or do organisms have when this goes wrong?
The major issues that people have probably heard about are things like Down syndrome, trisomy 21. That's an error in chromosome segregation. The other major error that people aren't quite aware is that the rate of spontaneous abortions is about 30%. So for most women, you don't even know those because they're very early on. And it turns out that in the woman's body, cells are arrested, their eggs are arrested at meiosis, stage in meiosis. There's a protein that we're very interested in, cohesin. It holds the chromosomes together. There's something that happens that's called cohesin fatigue, which means literally the protein, you work on proteins, the protein gets tired after 20 or 30 years and sitting in mom, and literally those chromosomes are unable to be held together, and then there's errors when they do segregation.
I see.
So the rate of spontaneous abortion is very high because of that protein failing. And of course, genetic instability is predisposes to many different types of cancers, and so that's another reason it's really important to sort of figure out how cells get it right.
Very cool. Dr. Bloom, thank you so much for having us in your lab and showing us around and teaching us exactly how it is that centromeres help separate the chromatins.
Thank you, Thomas.
All right, and maybe we'll get some of these scholars to visit you in person someday.
That would be great.
All right, thank you.
Yep, thank you.
Bye.